Good morning, students. Today we are going to see about chromatography. So, chromatography is an analytical technique which helps in the separation of the components, and it is one of the most important analytical procedure which is being carried out in the lab, which helps in separation of the compounds. If the mixture contains different compounds, this chromatography helps in separating the components according to their structure or according to their size. It helps in separating the components. So let's see one by one. So first we have to know about what is chromatography and how the separation process. I've already told you it helps in the separation of the mixture of compounds. So chromatography is a technique for separating a mixture into their components in order to identify, purify and to quantify the mixture or the components. What is the purpose of doing the chromatography? This chromatography helps in analyze the components, then identification of the components, purification of the components, and to quantify the components. What are the components which we got after the separation? We can help quantify the components. So it is a laboratory. What is a chromatography? Is a laboratory technique for the separation of the mixture. And how this process which is being carried out, it helps by the two phases. It is being done by the two phases, that is mobile phase and the stationary phase. The mixture is dissolved in a fluid called the mobile phase. So mixture means here it is a sample. So what is the sample which we want to separate? No, that sample will be mixed with the mobile phase. That will be in the liquid form. So that is called as a mobile phase, and which carries it through a structure holding the another material which is called as a stationary phase. That fluid which moves or flows on the another structure, and that is called as a stationary phase. So with the help of this mobile phase and stationary phase only, the separation of the samples will be taken place. The various constituents of the mixture travel at the different speeds, causing them to separate. So when the samples which is being mixed with the mobile phase, when it moves through the stationary phase, what happens? It travels in a different speeds. So which helps in making the samples to separate. For example, if the sample is too large, the size of the sample is too large, it takes time to travel, and it will take a more time. And if the sample in the mobile phase which is very small, it takes a it takes a minimum time to travel. So it takes a minimum time to travel and to get ended. So by the different speeds, we can able to separate the samples. The separation is based on the partitioning between the mobile and the stationary phase. So what are the uses? We have already seen in the first slide. So what are the uses of the chromatography? It helps to analyze, that is examine the mixture, its components and their relations to one another. What are the samples components which is present in the sample? No, here we can able to analyze, and we can able to find out the relation between one another. And next is to identify, which the separation which helps in identify the mixture or components based on the known components. So after the separation, we can able to easily identify the compounds which are present in the sample. Then next is a purification. So after the separation of the components, we can able to go for the isolation of the components. That is a purification of the components we can able to do, and this as this chromatography plays an important role or the first step in purification of the sample. Then the last one is the quantify. It helps to determine the amount or how much amount which is present in the sample. That is each sample component, how much it is present, we can able to calculate by using the chromatography. And in number of industries, this chromatography plays an important role. The first is in the pharmaceutical company, which in the pharmaceutical company they use the chromatography to determine the amount of each chemical which is present in the new product. Suppose if any new drugs, if they are making, no, that the new drug or many tests will be taken place, and we have to know how much samples in that particular new product, how much the components are there and what are the components are there, we have to find out. So here in the chromatography we can by using the chromatography we can able to find out the sample, that is a new drug, how much the components are there and what are the components are there, we can able to find out. In hospital, it helps to detect the blood or alcohol level in the patient's bloodstream. And in the law enforcement, it helps to compare the sample found at the crime scene to samples from the suspect. So in many films, we know how they'll be investigating by using this. No, they will take some samples from the crime scene, which from the samples they will come and compare with the samples from the suspect list they will take, and from that they can able to compare the samples. So they will be finding out the culprit. So here in this how this chromatography used here, it helps in the separation of the compounds. So it can be able to quantify. So here after quantification, we can able to easily identify the sample which is being taken from the suspect and how it is matches, it gives the result. Then environmental agencies. In environmental agencies, it helps to determine the level of the pollutants in the water supply. And in the manufacturing plant, that is in the chemical manufacturing, it helps to purify the chemical needed to make a product. These are all the uses of the chromatography. So we are going to see first one of the type, that is a paper chromatography. So chromatography is a method of separating, identifying various components in the mixture which are present in a small, even in a small trace quantity that the sample is present, we can able to separate and identify the sample. There are different types of chromatography, on which one of the types is a paper chromatography. This uses the stationary phase, that is the paper. Here the stationary phase, no, which the paper which acts as a stationary phase, and the mobile phase, it is the solvent, that is the liquid, and it is one of the most easily what we can say, it's a easy type of paper chromatography. As the solvent wicks up the paper by the capillary action, an equilibrium is established for each chemicals in the mixture. As the solvent, when we keep the paper inside the solvent, that solvent which speeds up and quickly absorbed by the paper. And when we are applying when we have applied the sample and the solvent reacts with each other and forms the chemical reaction in the mixture, which helps in the separation of the components. The principle is of the separation is mainly the partition. So first we have to know what is partition. So here in the picture we can able to understand the solute dissolved in the liquid phase, which is being coated on the surface of the solid support. So here in this that is samples which will be there in the fluid, that is a solvent. So here that solvent and the samples react with each other and they can be separated. So substances are distributed between the stationary and the mobile phase. Cellulose layers in the filter paper contains the moisture which acts as a stationary phase. So in the filter paper, no, they can absorb the solvent. So that acts as a stationary phase. Solvent, organic solvents buffer which can be used as a mobile phase. The developing solution travels up the stationary phase carrying the sample within it. So what happens when we keep the paper inside the buffer? What happens? The buffer, the paper which starts to absorb the buffer, which travels up the stationary phase, and already we will be introducing the sample in the paper. So what happens when the mobile phase comes and reacts with the sample? They start to travel up. They start to carry the sample with it. The components of the sample will separate readily accordingly to how strongly they absorb onto the stationary phase versus how readily they dissolve in the mobile phase. So accordingly to how strongly they absorb and how readily they dissolve in the mobile phase, the components starts to separate. And already I have discussed that large samples which takes more time to travel, whereas the small size samples, no, which easily which gets separated, and it separates easily. So these are all the components of the paper chromatography. The first is the choice of the filter paper. Whatman filter paper, which is being used for the purpose of the chromatography. In general, this paper contains 98 to 99% of the cellulose, and there are various grade and types of the paper which are available for the separation of the sample. And the next one is the preparation of the paper. The paper has to be prepared before we are using the chromatography. It has to be cut. The paper has to be cut in the desired shape and size depending upon the work which we are going to carry out. The starting line is marked on the paper with an ordinary pencil 5 centimeter from the bottom edge. So from the bottom edge we have to draw a line that is 5 centimeter above the bottom. By using the pencil, ordinary pencil, we have to draw a line, and all the starting line marks are made 2 centimeters apart from the each other. So on the line we have drawn, no, in that we have to mark a dot that is 2 centimeters apart from each other. And why we are doing that? In the dot we are going to introduce the sample. Application of the sample. How we are going to apply? The sample mixture to be separated is applied as a small spot on the origin. Already we have drawn the line. Now in that line we have to apply a small spot of the sample. The spot is dried on the filter paper and is placed in the developing chamber. And here the micropipette or glass can be used to introduce the sample on the spot. And here the capillary is used for the sample application. Solvents. The number of solvents we can use it in the paper chromatography. Some examples like ethyl alcohol and N-hexane. And the next one is the chromatographic chamber. The chromatographic chamber can be made of many materials like glass, plastics or stainless steels. And the glass tanks are preferred most and it has been used in the lab. They are available in various dimensional size depending upon the paper length and the development time. Development of the chromatogram. The paper is dipped in the solvent in such a manner that the spots will not dip completely in the solvent. So we have to place the paper inside the solvent, no, that the spot should not be completely immersed into the solvent. And as we start up this reaction, no, the solvent will rise up by the capillary reaction, and it is allowed to run 2/3 of the paper height for better and efficient result. So when the solvent starts to travel and reaches the 2/3 of the paper height, we have to stop the process. As the development is complete, paper is taken out of the chamber carefully. After the process is carried out fully, we have to take the paper out of the chamber. And this is the paper chromatography, and this is the glass paper chromatogram. And by using the lid, we can make the paper